Welcome back to Ramblings of a Mathwoman, a podcast where I talk about whatever shiny object happens to catch my attention for the week. At least that used to be my opening, but then lately I've been covering topics that are actually trending, such as the chess Grandmaster experiment or the recently deciphered Zodiac cipher, and even now with this current episode. Which I don't really feel bad about because I think it's not only entertaining to cover these topics while they're being talked about, but in cases like today's episode, it's actually important to make this information accessible to everyone. And not just physically accessible by providing the links with the information, but also digestible so that it's easy to understand what these links are saying. I had plans on talking about this at some point in the future, but then a few days ago, a friend of mine called me and wanted to chat a bit about some things that she's been thinking about regarding the COVID vaccine. Because she's in a medical field, she's in line to receive it if she wants it, but since the vaccine has been rolled out for emergency use, that means that they didn't get a chance to study it as thoroughly as they would have wanted to had the situation not become so critical so quickly. And while we were talking about it, we both kind of realized that we didn't know a whole lot about it. We just had some partial information here and there that we heard in passing, and we weren't even sure if that information was from reliable sources. So I decided after that conversation that this is definitely something I should educate myself on sooner rather than later. And I figured while I'm doing it already, I might as well share what I learned with everyone because I know that many people out there are probably having similar concerns. And this is an absolutely fair thing to be concerned about, by the way. I think there's a huge difference between being anti-vax and being cautious when it comes to vaccines being rolled out sooner than they normally would be. Now, because the health situation is what it is and it's so unique and different than anything we've ever had before, the FDA is doing something that they don't usually do, and they're releasing all the findings on this vaccine and they're making it public so that people can go through it on their own and make informed decisions on how to proceed. But I think, unfortunately, when an organization like this releases findings, if they want it to be complete and not have any important information missing out, this also means that the information they do provide is going to be pretty dense and probably hard to read for anybody who doesn't have a background in biology or who isn't willing to spend time to dig through like 10 different sources to pick out the important information. When I started looking over everything during the conversation with my friend, I saw a lot of words that aren't only specific to biology or chemistry, but also a little bit of statistics, pharmaceutical research, immunology, just things that if you don't know them off the top of your head, you can easily fall down a rabbit hole of definitions and then find it difficult to dig yourself back out and piece everything back together into a coherent sentence that you can understand. And this is probably the part where I should explain why I think I have the necessary knowledge to read and summarize all of these studies. I mentioned this in one of my previous episodes, which was mathematical modeling, but I'll mention it here again because I've noticed there are a lot of people with absolutely no background in any of the topics that I mentioned before, and they're giving their opinions about how you should feel on the vaccine, which they're obviously entitled to do, but as a listener, I think it's always good to know if you're getting your information from a credible source. So having said that, here's a little bit of information about me and who I am. As the podcast title suggests, I am a mathematician, but my passion was always in applying math to immunology and study of epidemics. So during my undergrad, I've spent years doing laboratory research at the Department of Medicine Research at my university. And during my graduate studies, I did my master's and a small part of my PhD research before I dropped out on the growth of the glioblastoma multiforme tumor. During this time, I also worked with another university's hepatology department where I was modeling the interaction between hepatitis B and hepatitis D viruses. And I also studied epidemiology on the side of my regular math coursework while I was in school. Now that I'm out of school, I work at a biotech company in an immunology and neuroscience department as a data scientist. Now, all of that isn't to say that I'm an expert on this topic or that I have some sort of insider knowledge because of where I work, but more to say that I have spent years, if nothing else, reading hundreds and hundreds of scientific publications, so I have had a lot of experience extracting the important information from them and understanding what it is that they're trying to say. I will not be making any assumptions on my own. I will just be presenting the data that Pfizer, the FDA, and the CDC have made available to the public. I also want to mention that I am by no means compelled to make this vaccine sound worse than it is because I work for a Pfizer competitor and I want to make them look bad, or to make it sound better than it is because I work for Pfizer itself and I want to make them look good. I have no horse in this race as far as any financial benefits go, and the only benefits that I do get from this is having this pandemic end as soon as possible. Also, just so no one is disappointed at the end, I will not at any point be telling you whether I think you should or shouldn't get the vaccine. I will just be providing information that is currently available so that you can make the best and most informed decision for yourself. And then finally, if I do feel the need to make some assumptions at any point, I will be very clear on what information comes from respectable published material and what is my educated deduction based on what I've read. I know that this disclaimer is kind of long and roughly twice as long as my other ones, but I feel like it's warranted seeing how this is regarding a current health crisis. So if someone were to misunderstand something as published results when it's really just my opinion, the consequences would be much greater than any misunderstanding from any of my previous episodes. So I apologize for making this drag on, but I feel like it was necessary. Okay, now moving on to the main course. Now, I want to avoid falling down that rabbit hole of definitions that I mentioned at the beginning, so I'm going to start with some of the basic processes of how a virus works and why a vaccine is such an effective method of helping us fight off these diseases. And for anybody who's already familiar with these things, I'm really sorry. I promise I won't drag on too long like my little disclaimer, but these things are really important for painting the full picture, so I kind of have to go over them. All right. As you probably know, unlike bacteria, a virus is a very simple organism. And by simple, I mean as far as other organisms are concerned. A virus is composed of two main parts, which are the genetic material, either DNA or RNA, depending on the type of virus, and the protein envelope that surrounds the genetic material. Because of this, where bacteria are actually cells and have more of a complex system that allows them to multiply on their own, a virus can't reproduce without the help of a host. It needs to use the parts that are found in the living organism's cell in order to replicate its genetic material, create more protein envelopes, and then keep on reproducing. A simplest rundown of a virus's lifespan is it infects the host, enters their cells, the DNA or the RNA creates what is called a messenger RNA, also called mRNA, and then all that mRNA does is it codes for those proteins that encase the genetic material. That's its purpose. It doesn't contain any information about making the DNA. It can't be reversed engineered to make the DNA or the RNA. It just codes for that external protein. And then once these proteins are formed, they can cover the newly replicated genetic material, which then makes more viral particles. And they exit the cell or sometimes lyse the cell, which means they break it apart so they can get out. And then they go on to infect and reproduce and just create that cycle. Those are the main parts of its lifespan, and that's really all you need to know for the sake of the rest of this story. Now, the way our immune system works to protect us is it creates antibodies that recognize certain parts of the protein envelope, and that's what they attack. So when you first get infected, your immune system goes to work to identify the threat. It produces an immune response, hopefully gets rid of the threat, and then it stores in its memory that specific protein section that it attacked so that in the future, if it sees something like that again, it doesn't have to start from scratch and build a response. It already has it ready to go. It's like a little Rolodex that it flips through, and it's great. And because of this Rolodex, it's why you're less likely to get a viral infection if you've already had it once before. Or why the second time you get it, it'll not be as bad or severe as the first time, most likely. And then on top of this, our body also has another step for getting rid of these infections, and that's creating what are called killer cells. And this is a response that doesn't have to wait to attack the virus while it's floating around outside of the cell, but instead, it can kill the cell that the virus is in so that it can stop it from replicating and creating thousands of more viral particles. And I know this sounds bad when you first hear it because just intuitively, you would hear what it does and think, you don't want something that kills your cells, right? That's bad. Like, you want your cells to live. But the response is actually very common, and it's what keeps all our cells healthy by eliminating the ones that could cause problems. Be it because of the viral infection, or because of cancer, or whatever. This is a very essential step in our immune system. It's kind of the equivalent of you having to get rid of a bedbug infestation by only killing individual ones when you see them walking around, instead of identifying where they are in the house and then getting rid of the infested things. If you do both of these approaches, you'll be rid of the infestation so much sooner. This is why both the immune system and the killer cells work together to help keep you healthy. But not to go off on one of my many famous tangents. This is all thanks to our immune system and its amazing memory. This is how it works, this is how we want it to work, and it's beautiful. Mwah, chef's kiss. However, there's a flip side to this. Having a disease once might make your interactions with it better in the future as far as your immune response is concerned, but it doesn't change the fact that while you're building this response and working to get rid of the infection, the virus is still wreaking havoc on your body. Even though your immune system might be able to fight it off, in many cases, this isn't before the virus had the chance to make you very sick, spread it to countless others, and possibly cause long-term damage to your body. As smart as our immune system is, and as much as it's constantly evolving to stay ahead of the infections, these viral particles are trying just as hard to outdo you, and they actually have a much higher mutation rate because they're so much simpler than our cells. So by the time you naturally create a response to one variation of the virus, they can come up with 5 different versions of their protein encasing that your system won't even recognize. And now it has to start all over again. And the faster your system is able to identify this threat and shut it down, the less time the virus has to multiply and create these mutations. And this is where vaccines come to play. There are several different variations of vaccines, depending on the pathogen that they're meant to treat, such as live vaccines, weakened, not live, but they all have the same method for how they work, which is they allow you to build this immune response that you need without you needing to get sick in order to do it. So a vaccine is just a virus that won't get you sick because, like I said, it's either not live or it's weakened, so it won't cause problems like the living regular virus would. And this introduction still lets your immune system see the threat and start building antibodies, but now you don't have to deal with the consequences of actually getting the disease. And to reiterate, while you're waiting for your immune response to act, it's not like you're just sick. These diseases can create lifelong damage before your body has a chance to stop them. Being able to build this response by eliminating the actual disease step is huge. Because of this, vaccines have actually helped so much that they have done more good for humanity, more so than any other medical advancement in history. And this isn't according to me, this is according to Harvard Health. Now, unfortunately, even with all of these benefits, there are still some downsides to the vaccination process, and that is that it can take quite a bit of time to develop. Isolating that one part that is key to triggering the immune response while making sure to omit anything that can cause a disease, it's really hard in itself. But then once you figure that part out, you have to still grow it, which takes time, and it's just a process that's not that quick at all. And then about 30 years ago, scientists start to wonder if there is a better way that this can be done, a way in which a vaccine can be developed faster. Because remember, being able to do this quickly doesn't only mean you get to help more people faster, but it also means that you give the virus less time to mutate into variations that might all need up ending its own separate antibody. And we really lucked out with the timing on this because a new approach for this has been studied since like the 90s, but it didn't really get ironed out until very recently, which is great because the fact that it can be created much faster, that means that the studies can move along faster and scientists can have results on a vaccine's performance way sooner than they would had they had to use the original methods of creating them. And this new method uses that mRNA that I already mentioned, and it uses this to trigger the immune system. So for anybody who stuck with me and listened to my little blurb about Biology 101, this is why I thought it was important. You're welcome. But the reason this method works so much better and faster than the old school method, which is what I'm going to call it from now on, just because it's easier, it's because instead of trying to find a different part of the virus that can be safely introduced into the system to get this desired immune response, while still not causing any damage, an mRNA vaccine only introduces the mRNA that the virus creates. So because it doesn't have any of the viral genetic material, it physically can't produce the virus so that it can make you sick. Because remember, you can't make the genetic material from an mRNA. it's not how it works. It only codes for the protein. So because it only creates this protein that would be found around the genetic material of the virus, your body can still recognize it and learn how to fight it off, and then store that information for later use in its little Rolodex. The mRNA can either be a complete sequence and create all the proteins that the virus uses, or in case of the COVID vaccine, it only creates one part that our immune system responds to, which is called the spike protein that is found on the outside of the COVID virus. So now this vaccine can be developed faster because instead of having to grow these particles and then separate them out, all of which takes a lot of time, since this mRNA can be pieced together in a lab, that time is cut down drastically. Not only that, but because of how the old school vaccines would have to be grown, people with egg allergies wouldn't be able to take them because those vaccines would need to be grown in eggs. This is not the case for mRNA. So the COVID vaccine is actually safe for people with egg allergies. And my source for this is the CDC site, which has all the information on the vaccine, which I will provide, obviously, as well as all the ingredients that are in the vaccine, just since we're on the topic of allergies. Not only that, but while the old school vaccines could mainly create just the immune response, the mRNA vaccines have actually shown to also create the immune system killer cells, which is a double strike as far as protection goes. I knew that mRNA vaccines were really interesting before, but when I started really looking into them for this episode, they're actually way more fascinating than I imagined. I hope that it was a sufficient introduction for what I'm going to talk about next. I feel like I covered all the important bits without, hopefully, being too boring for people who already knew it before they started listening to me and just came here so they can get the lowdown on the COVID vaccine research specifically. So everything that I talked about above, I know because I've studied it. But I did use a couple of resources when it came to the mRNA vaccine and how it works against COVID. And for that, I used both information on Pfizer's website and a link that is from Harvard Health. For this next part, though, I'm going to go off of an article that's been published in the NCBI journal, which stands for National Center for Biotechnology Information, and it's titled The Clinical Development Process of a Novel Vaccine, an Overview. And this one is really good as far as outlining the process that the vaccine goes through as it's being developed and all the requirements that it needs to hit in order to pass and move on to the next level. When you hear that this vaccine was rolled out before it had a chance to finish all of its trials, it helps so much to know what these trials are and where exactly it stopped. While I do work in the biotech field and am familiar with all these phrases and phases in context of drugs that I work on, I still needed to look over this because I don't work on any vaccines specifically, so there are some differences between drugs and vaccines that made me feel a little uncomfortable to just talk from experience without having something that outlines the process for vaccines specifically. And I think that these are the steps that are pretty important to know because while they may get shoved under the rug as unnecessary detail, they are what will tell you which steps were completed and what the requirements for completion are. While I totally understand why some people are being cautious as far as forming their opinion on the safety of the vaccine, the way things are being worded right now, it makes it sound like these scientists threw something together, said a prayer, and then just started testing on the public. And that's not even close to how these things work. So when the vaccine is being developed, or actually any drug really, there are 3 phases that it goes through. Sometimes 4, but 3 are really key. Phase 1 is the first step. I know, crazy, go figure. And this is the step where they take the vaccine from the lab and start testing it on the human subject. So this is the first time they're testing it on humans. And before it gets to this point, it has already passed all of the lab testing that it needs and has been approved to be tested on people. So phase 1 is generally tested on a small sample, and the purpose is to collect the immune response of the subjects and evaluate the safety of the vaccine. All volunteers have to be healthy. And this is where vaccines versus drug trials are different because vaccines are always meant to be used on healthy individuals since they are a preventative measure, whereas drugs are meant to cure a disease. So vaccines are intended for people without the infection and will therefore always be tested on people who do not have the infection. But I digress. All volunteers have to be healthy, have to have a naive immune system, and that doesn't mean that their immune system isn't smart. It just means that they don't already have an immunity to the thing that you are trying to test. And they also have to be at a low risk of acquiring this disease. By lower risk, I mean because they are either not physically exposed to it as much, because of how much they travel or because of their job, but they also have to have a lower risk based on the blood work and how susceptible they are to it biologically. This is done because you don't want to test the first rollout of a vaccine on people that are susceptible to catching the disease, because that would suck. So you slowly dip your toes and first see how people react to it by giving it to someone who is otherwise fine on all fronts. And then depending on how that first phase 1 trial goes, you slowly expand it depending on what group you're actually supposed to be targeting. For example, if the vaccine was meant for children, phase 1A would be tested on these healthy adults that are not in any risk of actually catching the disease. And then when that passes, it would move on to phase 1B, where you test in a lower age range, but still kids that are not as exposed to the disease, so they're not very likely to catch it. And this is how it's been done for all vaccines, like the malaria RTSS vaccine. It was first tested on healthy malaria-naive individuals that live in the US, where the disease isn't an issue. And then after those trials passed, it got tested on individuals who haven't been exposed yet, but live in areas that are at high risk for malaria, such as Mozambique, Tanzania, Kenya, and then whatever others. And then after that, 1 to 11-year-olds got tested. And then finally, after that passed, they moved on to infants. So it is set up in such a way that you try to carefully monitor risks. And if any do appear, you want to make sure that they first appear in people that are able to fight them off most effectively. While in this phase, the subjects will keep checking in with their site at whatever the predefined intervals are, be it every week, every 2 weeks, or whatever, depending on the study specifics. And then they will also have tests done at the start of the study, so before they receive any treatment, at certain visits when they come in, and also at the conclusion of the study. And by testing, I mean hematology, which is blood work, urinalysis, which is urine analysis, biochemistry, and a whole bunch of other things. So the scientists will keep track of all of these things from start to finish. They're not just giving these people an injection and then saying, let me know how you feel in a week. This is extensive, and there are values assigned to everything. It's not based on a feeling. It can all be measured. And in order for a vaccine to successfully pass phase 1 study, it must meet a 95% confidence interval. This means that if there are 100 subjects participating in this study and more than 5 of them experience unsatisfactory results, it doesn't pass, and you have to start from the beginning until you have 5 or less. So if it doesn't meet this requirement, you start from scratch. And this is in terms of both safety and the immune response. So it's not enough to say like, oh, it kind of seems to work. This is actually a really strict guideline that is heavily regulated by third parties. And during phase 1 for the COVID vaccine, they were testing 4 different variations, and this is when one was identified as the most effective, and that's the one that continued on with the testing. So for those of you that are curious, the COVID vaccine fully passed its phase 1 study. All right, let's look at phase 2. Now, during this period, the researcher wants to figure out what the optimal dosing for the vaccine and what the preparations are like. And again, evaluating the results based on both safety and the immune response which get triggered. Here, they also start to diversify their test subjects more by age, ethnicity, gender, presence of pre-existing antibodies, and possibly other things depending, again, on the substance that is being tested. This part of the study is also a much larger sample size than phase 1. And we're talking like hundreds to thousands of subjects, all distributed across different sites in different countries. This increase in sample size allows for even more accurate results when you look at that 95% confidence interval, which is a standard that, again, has to be met in order for it to move on to the next stage. There are other details unique to this phase, but those are actually irrelevant details, and they all depend on different types of compounds that are being tested. So if anybody is super curious, I would say you should definitely read the article. But as far as information that is necessary to understand a COVID vaccine, I'm just going to leave it at what I already said. The most important part that is figured out in phase 2 is testing dosage, mixing, safety, and efficacy on a larger and more diverse group of volunteers. And just like in phase 1, if you don't meet that 95% confidence interval, you go back and you try again. And again, for anybody keeping track, The COVID vaccine also passed its phase 2 trials with flying colors. So far so good. And now on to phase 3, which is the final one. And by final one, I don't mean that in terms of after this phase is completed, everyone stops working on this vaccine and then moves on to other things. But I mean it in terms of after this step is completed, you can file for FDA approval for a license to market the product. And remember, the COVID vaccine made it to this phase, even not considering the emergency use scenario. So for phase 3, the number of volunteers is even larger than for phase 2, like several thousand people all over the world. And this phase is also kind of tricky to explain because the details of how you proceed with testing greatly depend on the compound that is being tested. So it varies a lot from case to case. For example, for most studies, you want to be able to have a test and a control group, and maybe even another group that is receiving another form of medication. That way you have your substance that you're testing, a placebo group, and you still test it against another compound on the market. So you can compare the data you receive against several factors, which makes it that much stronger. But the problem is that even though that makes for a stronger experiment, in some situations, that's pretty unethical. If someone has to go out and be exposed to a pathogen and you give them placebo without telling them just to see how they react, and then they go out and get infected while they're thinking they're safe, if it's a deadly disease or something that causes permanent damage, you don't really want to do that. This is immoral. It just shouldn't be done, and luckily it can't. So in some cases, you may not be able to have a placebo group. Because of situations like this, from here the specifics of the approach stratify a lot. But there are still certain key things that are done in all cases, and those are the really important ones here. This stage is where they evaluate the vaccine efficacy by looking at the exposed individuals that are vaccinated and the exposed individuals that are unvaccinated, and then they calculate the value which is called vaccine efficacy. So how effective is this vaccine? They also look at the indirect effects a vaccination has on an unvaccinated population, which seems like it doesn't make sense, but if a vaccinated person is now less likely to spread their disease onto others, then the unvaccinated population is still benefiting from this even though they didn't get vaccinated. On the flip side, if the vaccinated population feels great and gets better really fast because of the injection, but they're still contagious while they're running around thinking they're fine, then this affects the unvaccinated population negatively because this probably increases the number of sick people walking around and spreading their germs. So the unvaccinated now are more likely to get infected than they were before. So these things are important to consider. They also look at any serious adverse events that occur in any of the patients, how the vaccine interacts with any other medication or supplements that they might be having, if people with any specific conditions or prior procedures or surgeries have a pattern of having certain responses that are either positive or negative. This step is where all the kinks have to get addressed, and they really do look at everything you can think of. Before it's released for emergency use, the COVID vaccine finished its preliminary phase 3 stage. It had the necessary 95% rating in terms of being effective against COVID across age, gender, race, and I think BMI demographics, and over 94% efficacy in individuals that are 65 years of age or older. Out of 43,000 participants, no serious side effects were reported aside from 2% of people having headaches after injection and 3.8% reporting fatigue. And to be clear, this means that they are temporary side effects that they experienced right after injection, not that they are now permanently fatigued or have lifelong headaches. There were also other minor side effects such as allergic reactions that you may have heard about on the news, but they didn't last that long and all ended up resolving themselves without the need for any medical intervention. So this all sounds really good, right? And you're probably wondering why it's been rolled out for quote-unquote emergency use if it completed its phase 3 trial. Like, it should just be ready to go, right? And this is what I was talking about at the beginning, where if these processes are better understood by the public and they knew which ones the COVID vaccine had already passed, they would be way less skeptical. It takes it from just seeing it as an incomplete experiment to an experiment that is mostly done, but there are still some questions that it would be useful to have answers to. And that's kind of where the COVID left off. Most of the parts are completed, but some questions are still up in the air, and that's what is currently being worked on. So even though all these important things have already been tested and passed the same way they would need to in a non-emergency situation, data is still being collected so that we can have as much information as possible. For example, our immune system memory, that little Rolodex, whether it's created by a vaccine or exposure because of the actual infection, it's not always lifelong. At the moment, researchers aren't sure how long the immunity from the COVID vaccine lasts. It could be permanent, it could be a few years. The study isn't concluded until we know this, so this is still an open question. Another thing that is uncertain at the moment is whether people who receive the vaccine can still spread the disease. And this is one of the important things that are mentioned as part of phase 3. So that one didn't get the chance to be fully concluded yet, which is why you might hear doctors say that even though you got the vaccine, you should still wear your mask. And this is because even though you're much safer with a vaccine, you could still be putting others at risk, and we don't want that. Another thing that needs to be studied is the vaccine has to be administered in 2 doses, which are 3 weeks apart. Does just 1 dose work? It does provide partial immunity, they concluded from the studies, but how long does this partial immunity last? Is it useless unless you get both? At the moment, there isn't a clear answer. So these things that are mentioned are just some questions that are left over that make this experiment incomplete. But they're definitely not something that should raise public concerns over whether or not the vaccine is safe. And this is information that the FDA, CDC, and even Pfizer have made public so that people can read it and understand. The only real downside is that this information is distributed across several journals and articles, some of them not that user-friendly, containing non-trivial vocabulary, and all these things combined make it so that most people, even if they manage to find this information, won't get a chance to read and digest it all. Which is why I wanted to make this episode and take out the relevant bits and kind of make it more accessible, because I think this is the information that everyone should have available. This is what gives you the power to make informed decisions. If you go to the Pfizer link, you can find the exact distribution of people who received the first dose, the second dose, different ages, ethnicities, and how many volunteers were in each of these categories. I just think that the stats are really nice for people who actually care about these things because you kind of get the bigger picture. It paints the whole story for you. This is all the information that I found useful for anybody who might consider getting it but is still torn. And while I did say at the beginning that I'm not going to tell you whether or not you should take it or not, I will say that when I'm given the chance, I will take the vaccine. So do with that information whatever you will. So that's basically the rundown. This is the most accurate information that's out there as of January 10th, and I might do an updated episode once more information is known if I find it's important enough to talk about. This is all just my educated opinions based on the sources that I had available, and I will always encourage anybody to look into these things themselves before making any decisions. While I did list my credentials and I stand behind what I said, I am still just a stranger with a platform, and I think you should always take that with a grain of salt, no matter who they say they are. Okay, conclusion time. And this time I do have a conclusion to wrap this whole thing up with, but it's not really about the vaccine topic specifically. It's more about how this type of partial information, like the one that I mentioned in the beginning, can end up leading to a complete misunderstanding of what the reality actually is. And that's actually another topic I assumed I would have to talk about at some point in the future, but since this is relevant now, I might as well just cover it. When it comes to science and scientific experiments, and even in my job as a data scientist, a researcher's duty doesn't end at the conclusion of the experiment. How the results are interpreted and presented is just as important as the result of the actual experiment. I'll even go as far to say that I would prefer to take a poorly executed experiment that is well documented over a perfectly executed experiment that doesn't share full information about what was found at the end. And I realize that in the case of the vaccine, I'm not blaming the people who did the research. It's more that the issue of how the news was spread through the media, but I still think the point that I'm trying to make stands. To give one example of what I'm talking about, I remember a while ago that I was reading something that said that flossing doesn't prevent cavities. And there's actual research done on this, and because of this, there's a bunch of articles out there saying that there's no proof that flossing has any benefit because there was no noticed correlation between people who floss regularly and the amount of cavities they get. And when I read this, it kind of blew my mind because it just didn't feel like it made any sense from my experience with flossing. So I talked to that friend that I mentioned at the beginning of my episode because she's about to graduate from dental school and she really knows her stuff. And I asked her what the deal was with this whole flossing thing. And she kind of sighed and rolled her eyes because apparently she has gotten this question quite a few times from different people. And she told me that it's true. Flossing doesn't prevent cavities, but it does prevent gingivitis, which is just as important when it comes to the health of your teeth. So if your gums are in bad shape and they start to deteriorate, your teeth won't have much of a chance even if they are cavity-free. But to me, this was a prime example of true yet partial information, and as a result, no matter how true the bit that was shared was, it was still incorrect. Yes, flossing did not prevent cavities. That was true. But the result that was extracted as a result of that partial information was really incorrect. You should still continue to floss. Another example was brought up during a discussion when someone was trying to convince me that going to college was a waste of time. So he brought up a statistic that I'm pretty sure it was true maybe like 40 years ago, but it's outdated now. But he said that there are many more people who are successful that didn't go to college than people who did. And according to that statistics, college was a waste of time. When you read a result that's worded like that, it's very easy to make a conclusion that he did. And I'm sure a lot of people have come to the same result. But what the statistics should have pointed out was that there were also way more people who didn't go to college than people who did. So while there were more successful people without these degrees, there was still a higher percentage of people who were becoming successful coming out of college. So if you're playing the numbers game, your chances are higher in one group over another. Again, I don't think that statistic holds anymore, but I stand by my point about the data interpretation regardless. But the whole point of this entire ramble was that I honestly see this type of information sharing on the same level as lying by omission. And that's why I said that a researcher's job doesn't end at the conclusion of the experiment. And you kind of have to keep an eye on these results that you put out into the world and make sure that they're not getting misinterpreted in dangerous ways, especially when it comes to medical things. If you do, then things can easily spiral into mass hysteria, and then from that, it's really hard to come back. So it's much better to be able to just nip the whole thing in the bud as soon as you can, which I guess is what the point of this episode is. For anyone who's listening to this for the second time, first off, thanks. But second off, you probably noticed that I changed my conclusion a bit to include the results interpretation bit and that I took out something else that I talked about. I still like the point that I made earlier about the potential philosophy or why people are so hesitant to get vaccinated or to take certain medication, but I feel like the actual episode had plenty of information in it already, and I don't want to overload it with any extra unnecessary things, so I'll probably just circle back to that later with some other episode when I feel like it's appropriate to fit it in. Alright, thank you so much for listening. I really hope that this helped out a bit and that it provided a bit more information that you can find useful. Feel free to reach out to me on my Instagram, Ramblings of a Mathwoman, and I'll talk to you next time. Bye.